Momo, would you mind just sharing who you are and what you do?
So my name is Momo Vuyisich, and I am the Chief Science Officer for Viome, and I lead our efforts in product development and clinical research.
What's your background?
I'm a biochemist by training and had a lot of different kinds of scientific efforts over a pretty long career before founding Viome.
You're very humble because you're world's one of the most, expert on gene expression.
Well, it's just one of the tools in the tool chest. So yeah, we're using some of the most advanced gene expression tools at Viome.
Can you just explain what's the really difference between the RNA and the DNA?
Yeah, I think that's a very important issue to understand. So DNA is a physical object that's found in all of our cells, and it has a potential for a function. However, when a gene needs to perform that function, it first has to be copied into RNA molecules. And so without RNA molecules, there's no life and there's no functions. And so our cells copy the gene into RNA, which is then translated into function. And that's what we actually see in our physiology.
So just to be clear, DNA tells you everything that can happen, and so it's like an alphabet, and RNA is actually what is happening. So if whether it's writing a poetry or a thriller, so DNA is an alphabet and RNA is actually what is being done with that.
Yeah, I like that metaphor.
Okay, so as you look at this DNA and RNA, why is it important for us to look at RNA versus DNA, especially when it comes to chronic diseases?
So let's think of a chronic disease. For example, rheumatoid arthritis. Many people have that disease. Millions of people have that disease. Rheumatoid arthritis comes and goes, meaning that people will be in remission and will be essentially symptom-free for months or years. And then they'll have a relapse where they'll suffer very bad symptoms for months or years. And no one really understands why that happens. And it goes through these cycles where cumulatively there's joint damage that accrues over a period of years. It's clear that there's very little genetics that plays a role in these changes going from remission to relapse. So it's all about gene expression. It's all about inflammatory response to some things, to some environmental stimulus that we don't yet understand, and that changes the disease. So it's really all about gene expression and not genetics.
So why is it that everybody is really looking at DNA and they think somehow that looking at the DNA will give them some idea about why you are sick? And what I'm hearing from you is really that it's not like you suddenly have a disease that's flaring up and you say, Mom, my DNA changed.
That's exactly right. So I think the main reason why almost everyone is studying DNA is because it's easier. DNA is a very stable molecule. If you leave it in a tube on the bench for 2 weeks, it's still going to be there. RNA is a very unstable molecule. It is constantly produced and constantly degraded in cells, in living cells. And for that reason, it's a far more informative molecule. So I'll give you one example in the microbiome space, in the gut microbiome space. We've all heard how the gut microbiome has millions of genes and how they outnumber our own genes by hundreds to one and so on. Of those millions of genes that are encoded by our gut microbiome, there are only a small fraction that are actually expressed and play a role in our physiology, meaning our health and disease. And it's really understanding those expressed genes, the expressed functions of the gut microbiome, that is important in our understanding what gut microbiome functions lead to health and disease.
Interestingly, the technology that you're talking about that Viome uses, where did it come from?
So this was developed at Los Alamos National Laboratory. It was funded by the US government in order to develop one capability that can identify every single living organism in any sample. And the only way to do that is really to analyze RNA. And so the government didn't want to study separate organisms and try to guess as to what might be in a particular sample. You simply want to have one test, and in that one test, you can identify every single living organism on this planet.
However, it's not about just identifying the organism. The purpose really was to find out what they're actually producing, because this is really what they needed to know to find out if something bad happens, what is making people sick.
Exactly. So you've heard of these strains of bacteria that can cause disease, but they also live on us or inside of us. So for example, Staph aureus is a known pathogen. However, Staph aureus lives on us as a commensal, as a friendly organism, but sometimes it turns into a pathogenic organism. So if you look at this organism from the DNA perspective, it's identical. It's the exact same organism. You can sequence it 1,000,000 times and you'll get the exact same data back. However, from the RNA perspective, when it's a friendly and commensal organism, it's expressing certain genes that enable it to live in our throats, for example, as a friend. But when it starts expressing these virulent genes, that's when it becomes pathogenic. And the only difference between a pathogenic Staph aureus and a commensal Staph aureus is in gene expression.
By looking at the gene expression, we can find out essentially what is being produced and what may be causing the disease. And then once you understand the biochemical activities, you actually, at that point, it's just simply the chemistry, and now you can apply the right set of math to it and to be able to solve the problem.
Yeah, so I think this is a really important concept. So we have all read about bad bugs and good bugs, right? In actuality, most of the microbes in our gut microbiome are able to produce bad chemicals, and they're also able to produce good chemicals. And so let's take a...
Hang on for just right there. So this idea of, oh, I am taking these good probiotics that are good bacteria and your bad bacteria, you're saying that all that is really not true, that anything like a human being can be good or bad given in a certain situation.
Exactly. Just like a human being can be good or bad, it's the same person can be good or bad depending on the function. And so going to probiotics have been shown to have good effects towards certain symptoms, but also some people take them and they have bad effects. And so the same exact probiotic in one person can have good effects in another person, or under different circumstances have good effects. So let's take a very, very simplified version. Let's say that one organism, just one simple microorganism that lives in your gut, is able to produce pro-inflammatory biochemicals. They activate your immune system unnecessarily, and your immune system starts to develop inflammation. But they're also, that microorganism is able to produce an anti-inflammatory biochemicals. These are biochemicals that calm down your immune system, and there is no damage done to your body. For each one of these functions, that microorganism needs food in order to activate those pathways. And so depending on which food you feed that microorganism, it can either produce one of those biochemicals, the other one, none of them, or both of them.
So hang on for a second here. You're saying just if people were just looking at what organisms are there, they would never know that actually they may be behaving completely differently based on what you are eating. So what you're eating changes them, and that means now you can use this knowledge to tell people what they should be eating for these people to do the right thing for you.
Exactly. So that same organism that I just gave you an example of, is it bad or is it good? It's neither. It can be good or bad depending on what biochemicals it's producing. And the biochemicals that it's producing solely depend on the food you're ingesting, because that microorganism cannot produce these pro-inflammatory chemicals just by itself. It can't produce them from thin air. It actually consumes the food you ingest and specific foods and converts them into pro-inflammatory chemicals. And then it converts other foods into anti-inflammatory chemicals. So because we have that knowledge and we can measure those pathways, we can tell that person, because they have that organism, exactly which foods to eat in order to avoid the production of pro-inflammatory chemicals and to increase production of anti-inflammatory chemicals.
So Momo, I want to now take just a step back. A customer gets the Viome test. They send them the most precious thing they have, their poop, to you. Now I want to see the journey of this poop into the things that they see, what food to eat. Can you share this background of what happens to my poop that goes in there?
The poopy journey. Okay, so yeah, this is an interesting thing. So first what happens is that poop is preserved in our special preservative that freezes this RNA molecule the way exactly it was when it was produced. That way, during shipment, there's no degradation of these RNA molecules. So this sample arrives at our labs, and the first step that we do is we crack open all of the microbes. So the technical term for that is cell lysis. So we physically physically crack open every single microbe in the sample, and then that's how they spill their RNA content into the solution. And now we take those RNAs, which there are billions of from every single one of those organisms, and we
Hold for a second. The tiny poop that I sent you had billions of those things?
Many, many billions, yes. The molecules are incredibly small, so billions of them. So we don't want to actually sequence all of them because 99% of them are non-informative. They're called ribosomal RNAs, and they're just basically occupying the space there, and we don't want to sequence them because we already know their sequences, and so they're non-informative. So we actually fish them out physically from the sample, and now that 1% that remains, we sequence that 1%. So we identify the actual sequence of A's, C's, G's, and U's in this case. So RNA has a U instead of a T, but it's the equivalent of a T in DNA. And we sequence all of those, and we actually sequence 10 million of those molecules for each stool sample that we get from our customer. 10 million.
You mean 10 million times?
We sequence 10 million molecules. Different molecules randomly from the pool, and they represent the microbial community that lived in the poop.
Wow.
And so now we have very, very fancy bioinformatic analysis that analyzes every single one of those molecules individually. So we not only identify which organism that molecule came from, but we know which gene it came from and what function was encoded by that gene. So not only do we know which organisms are there at the strain level, we now can identify and quantify the gene expression of all the microbes in the poop down to the level of 1 in a million. So if a gene is expressed at the level of 1 in a million or higher, we can actually identify and quantify its function.
That is unbelievable. 1 in a million, right? Wow. So keep going. So what happens to my poop now?
So now that we've identified which genes are expressed by the microbes, the work continues in putting those genes together into pathways. So remember, I just talked to you about how a microbe can use some food molecules from our food, right, and convert them into, let's say, pro-inflammatory biochemicals. That's called a pathway, an enzymatic pathway. So it's a system of enzymes which come from genes that convert that food molecule into a pro-inflammatory molecule that causes inflammation. And so we actually put together several genes into a pathway, and we quantify the activity of that pathway. And that's how we measure. We don't infer, and we don't have models for that. We actually measure the activity of these microbes, and that's how we have this inflammatory score that tells every customer how high their gut microbiome-induced inflammation is.
And then continue. So what happened? How do I get my recommendations now?
Okay, so now let's go back to that simplified example. So that one microbe that is able to produce both good and bad compounds, pro-inflammatory and anti-inflammatory, we now know exactly which substrates, which foods it's using to produce pro-inflammatory compounds, and our computer model will immediately place those foods into minimize and avoid. And then that same microbe, remember, same exact microbe can produce anti-inflammatory compounds, and we now know which foods it uses in order to produce those chemicals, and we want it to produce those chemicals, so we want to feed it those foods. And so now foods containing those particular molecules are actually placed into the enjoy and indulge list, which means that we want to feed the microbiome those foods so that it can produce anti-inflammatory biochemicals. And that's how we fine-tune the effects of gut microbiome on our physiology. We give it exactly the molecular foods that it needs in order to produce beneficial and not to produce detrimental chemicals.
And that's how we know that when I say don't eat these food, we tell you why you shouldn't be eating this food. And if we tell you to eat a certain food, we tell you why you should be eating certain food. All this magical stuff that you do there is all from a tiny poop. So my poop is the most important thing is out there.
It's far more important than our genes, right? Our genes, at best, will tell you that you have an increased risk of something. Let's say you have an increased risk of depression, but you don't have depression. Do you care about that risk? You don't. And if you had depression, you'd say, well, I have depression. That doesn't help me the fact that I had a higher risk. I want to get rid of it. This way, we're actually fine-tuning the biochemical output of the gut microbiome, and eventually we'll be able to treat diseases using this approach.
So I am really curious now, Momo. If my poop, if I'm healthy, and when I'm healthy, my poop is like gold. Somebody wants it because somehow that is the most important thing. When someone is sick, they can take my poop and cure themselves. That is it's like a gold if I could save it. there are certain people who can eat bread and never get fat. Wouldn't their poop be so valuable, more than even gold?
No, I don't think so. So I think that this concept of bad and good microbiome or healthy and unhealthy microbiome really needs to go away. What we are finding out is that almost anyone's gut microbiome can be healthy. All we have to do is match that microbiome with the personalized diet for that person, and their microbiome automatically becomes healthy, just like based on the example I gave you. So
you're telling me I'm going to make no money from my poop anymore?
Everyone has golden poop. Everyone.
Oh, God. I was thinking of store my poop and start charging people for it. So that's gone.
No, you can't do that. But let's talk about the magic part. Okay. So I really want to mention that. So yes, until now, poop and diet and trying to find a diet for your condition really has been magic. And the reason is because we don't understand it, right? If someone flew a helicopter in 1800s into a town, people would consider that to be magic. That's not possible, right? And that's not because it's anything magical, it's just that we didn't understand it at that time. For the first time at Viome, we are now making personalized diet recommendations based on chemistry of the gut microbiome and mathematical equations that tie the substrates found in foods and the products of those biochemical pathways that affect our physiology. So we've converted this sort of guesswork of, is it keto today? Is it Mediterranean tomorrow? Today coffee is bad for you, tomorrow's coffee bad for you, right? Every single day there's conflicting dieting information. Instead of that, we actually bring chemistry and math to your poop, and based on your gut microbiome, we can make very personalized diet recommendations.
So essentially, this is a technology that is so advanced, it's so magical, it is so easy now, that, it seems like a magic, but it's really just a simple science.
Well, it's simple science, but it's actually very complicated in that there are thousands of biochemical pathways in the gut microbiome that we analyze and quantify, so it's very complex. But we have machine learning that can analyze this vast amounts of data and learn these rules and apply them to every single new customer. So we've made it simple, but it's really not simple.
So Momo, it's amazing that, we're learning so much, and that's how we are essentially taking something that was mythical and making it into magical science. Now, interesting thing is, now we have had 100,000 plus people that have taken the test. As these people are coming along, what have we learned, and where do you go from here?
Yeah. First, let me say that instead of calling it a magical science, let's call it objective science. It's very objectified. So today, when a customer provides their stool sample to Viome, there are no opinions, there are no people's experiences, there are no anecdotes. What happens is the data from the poop sample are actually fed into machine-learned algorithms, and they produce personalized diet recommendations. It's 100% objective. There's no human involvement, so that's great. Okay, now that we've analyzed all of these samples from all of our customers, and most importantly from our clinical research studies, we can now start to see and form very objective ideas of what these chronic diseases actually are. And so, for example, let's take depression. No one really understands today what causes depression. They have found some genes that are correlated with depression, but then those correlations were actually refuted. So genetics doesn't seem to play a role in depression at all. And so currently there are many medications that treat depression, but no one really understands or tries to understand the root cause. It turns out that based on a lot of data that we've gathered, we can actually determine whether someone has depression or not with 85% accuracy just based on the stool test analysis, right?
I'm going to repeat yourself. You're telling me just by looking at my poop, you can tell me if I have a depression?
If you're depressed with 85% accuracy without knowing you or seeing you. So the determinants of depression are not some magic. It's not something abstract. It's literally the biochemicals produced by the gut microbiome, or in many cases, lack of chemical production by the gut microbiome. And so now that we understand what we call a disease signature, right? We understand what the gut microbiome of people with depression is producing that healthy people's gut microbiome is not producing. And then very importantly, what is the gut microbiome of depressed people not producing that in healthy people it is producing. Now we can use those mathematical and chemical equations to actually personalize the diets and try to reverse and prevent depression. And we're going to do that in clinical studies where we're going to show efficacy of this approach, where we're going to recommend personalized diets to people and compare the efficacy of personalized diets versus placebo in a clinical trial format.
So Momo, what I'm hearing from you is that you are now able to not only understand that somebody has depression, now that you know what makes the depressed people depressed, you can prevent it, but moreover, you can actually reverse it now.
Exactly. We are going to demonstrate that in a prospective clinical study, but yes, I'm very convinced that we can do this because we can manipulate the biochemical output of people's gut microbiome by either providing them with certain foods or denying their gut microbiome certain other foods.
But is it just depression, or could it be applied to other diseases?
Every single chronic disease that we've had enough people participate in these studies for, we have found this sort of a signature. So for now, we're very focused on the big 4 diseases. So these are depression and IBS, and obesity and diabetes. So these 4 chronic diseases make up more than 50% prevalence in the world's population. So more than 50% of the people suffer from one of these 4 diseases. And we have very strong biochemical signatures in the stool of people with this disease. And now we're going to address those at the biochemical level and try to reverse them and or prevent them.
So, Momo, is it fair to say that in next 5 years, just look 5 years, we were going to have a world where these diseases may not even happen, or we'll be able to actually cure them, not just suppress them?
I would say 5 years is unnecessary. I think we're going to be there much sooner than 5 years. I would guess in 2 years we'll be there.
Really? And what about the things like, obviously you know that I lost my dad to cancer. It's personal. I mean, do you think we'll be able to find the cancer, diagnose the cancer, to be able to reverse the cancer?
Yeah, so gut microbiome has been shown recently to be involved in maintenance and potentially cause many of the cancers. And in particular, pancreatic cancer was shown recently to be actually caused by the gut microbiome in a mouse model. And so in this particular case, specific members of the gut microbiome would travel up the bile duct into pancreas and then would essentially set up shop there and say, wow, there's plenty of food here. All we have to do is shut down the immune system of the person. And so those particular microbes are able to shut down the immune system in pancreas. And because our immune system patrols the entire body and kills cancer cells when they arise, this inability to get rid of cancer cells is what allows cancer to arise in pancreas. And so this is possibly a common mechanism for many of the other cancers, because there have been already publications showing that there is microbiome present in most tumors that have been sequenced. So this could be a universal mechanism that we can not only identify, but prevent.
So, Momo, so what I'm hearing from you is it could be cancer, especially pancreatic cancer. What are other cancers you think there may be a good likelihood that we'll be able to?
Colorectal cancer is absolutely for sure contributed to by the gut microbiome, so we are going to make good progress there. But breast cancer as well, ovarian cancer as well, and so uterine cancer as well. So we'll get to these cancers, but that's sort of the next tier of diseases that is going to take a little bit longer to understand and solve. So maybe for those, we're talking about 2 to 4 or 5 years.
So you're saying in the next 5 years, if you just look out, you feel hopeful for the humanity that there will be a day in 5 years where we'll be able to prevent or reverse cancer, prevent and reverse things like depression, diabetes, obesity, IBS, and even other chronic diseases, I suppose.
Absolutely. I think all of these chronic diseases, because there's no genetic component, if you studied, twins have been studied for all these chronic diseases, and it's clear that the genetic component is not strong. All of these chronic diseases are likely caused and or modulated by the gut microbiome and our diet.
I'm going to ask you the 2 common things that everybody worries about as we start to get old: Alzheimer and Parkinson dementia, or MS. All these are kind of chronic diseases that we have a shot at someday not having?
Absolutely, yes. So specifically for Alzheimer's and Parkinson's, it's already been shown that there's microbiome activity in the brain that is likely causing the disease, or at least some portions of it. And so we just need to figure out what the mechanisms are and what pathways lead to this presence of microbes in the brain and lead to these diseases.
I mean, Momo, this is probably one of the most exciting field, if you ask me, because there is no better way to live a life than to be working on something that could, in the next 5 years, now you're not talking about even 10 years here, right? In the next half a decade, we could fundamentally change the way people are going to live their life without disease, a world with no chronic diseases.
Yeah, this is very, very exciting. So what's really exciting is that people who wanted to solve this problem 10 or 20 years ago, they simply couldn't because the technology wasn't there. And now, luckily at Viome, we have all the technology, both experimental on the lab side and computational, to actually solve these problems. And so we are going to solve these problems very quickly here, and we're very excited to be leading the charge.
So Momo, a lot of the times people say, I am healthy, I'm just good. I don't see the rashes on my face. Why should I be doing this Viome test? I'm young, I'm healthy. Do I really need this?
If we look at all chronic diseases, they don't happen overnight. The pre-symptomatic phase of these diseases lasts years or decades. So for example, no one eats a steak one evening and gets a heart attack the next morning. No one drinks a soda today and gets diabetes tomorrow. These are chronic diseases and they take years and decades to become visible to us. And so what Viome offers is the ability to look into the molecular processes. Basically, we can look under the hood and make sure that your gut microbiome is actually producing healthy biochemicals and it's not producing detrimental or harmful biochemicals. So we can fine-tune the gut microbiome of everyone and hopefully keep them healthy forever.
So you're essentially saying you're looking for the rashes inside my body, that essentially you can show me what's happening inside. So even though I don't feel anything, I don't see anything, but actually I'm able to see inside the body at a biochemical level what is going on. So I can actually stop it even before I see the symptom.
Exactly. I mean, inflammation that leads to a heart attack or some other inflammatory disease like obesity or diabetes, this precedes the actual symptoms by years or decades. And so why not intervene before the disease symptoms arrive? You can intervene years or decades earlier by eating exactly what your body needs and avoiding foods that your body should not be eating.
Momo, it's amazing that people talk about you eating food and it changes your microbiome. How long does it really take? I mean, am I worried that I take a bad meal last night, I shouldn't be doing this test? And when is a good time for me to be sending you my poop? Because, that's not, I'm worried about that. What are you going to find out? Should I be waiting for eating healthy for a couple of months before I send you the poop? And how long does it really take after I follow your recommendations, when I'm going to start seeing the impact of it?
When it comes to sample collection, it doesn't really matter what you ate the day before or 3 days before. Just collect your sample and send it to us, and it's going to be reflective of what your gut microbiome has been looking like for the last couple of weeks. We already have published data to show that the gut microbiome is relatively stable over a period of a couple of weeks. So to answer your second part of the question is how often should people retest? We generally recommend that people retest every 90 days. And the reason is because when you start following the new Viome recommendations, then you're going to induce changes in the gut microbiome such that in the next 90 days, the microbiome will change. And then we recommend that you retest so that you can get the diet recommendations now that fit your new gut microbiome.
So it's like you're saying, it's like a seesaw. You get a new diet, you start to balance, and if you don't stop, you're going to get the imbalance again. So it really is after 90 days or so, you should retest and get a new diet so you don't essentially go imbalance again.
Sure. Another metaphor that I like is simply like maintaining your car. Just because you changed your oil today doesn't mean it's going to last forever. Your engine is going to run forever. Simply do it on a regular basis and make sure that you're providing your gut microbiome that you have at that moment with the best foods so that it produces only the beneficial chemicals and it does not produce harmful biochemicals.
If I have just taken antibiotics, can I still send you the test? How long should I wait if I've really taken antibiotics recently?
So it depends on the person, but in general, I'd say wait 1 week from the moment you stop taking antibiotics, and your microbiome will essentially establish itself after a week, and then you can collect a sample and send it to Viome.
But if I'm traveling on a conference or something, it's not that I need to wait until I come back for a week and start eating my food normally?
No, absolutely not. The microbiome is stable unless there's a major perturbance like antibiotics.
So Momo, people hear about these diet recommendations, and there are a lot of skeptics out there and say, Weight Watchers told me to eat this, and my friend had a keto diet and, he really helped lose the weight, or someone says my friend has been on a paleo diet, or this intermittent fasting. And all these fad diets come and go. Aren't you just another fad diet here, or what's going on here?
Well, we're not a fad diet for the main reason that we're personalized and we're basing the diet recommendations on your gut microbiome. If there was one diet that was best for everyone, it would have been found by now, because millions of people have tried different diets. So that's one thing. The second thing that I want to mention is that other diets that even may be based on gut microbiome tests, they're based on literature references that use maybe 10 or 20 participants. And so these small studies are the reason why we get conflicting food advice. So one study says coffee is bad for you. Another study says coffee is good for you, right? And the reason is because the few people that each study has chosen for random purposes have either a good or a bad effect, if the coffee has a good or bad effect on them. What we've done is, the 4 big studies that I mentioned, where we looked at 4 big diseases, the minimum number of participants in each one of those is 1,000. So we are 2 orders of magnitude above other published studies. And we have this test that not only tells us who is roughly there, we actually have a test that tells us exactly what each person's gut microbiome is doing. So we have hundreds of times more information that's actually relevant to human health than all those other publications.
If I am the person who takes a test today, and is it like if I somehow don't follow it, have I committed a sin? And is it like how should I really look at this diet? Because there's a time I'm not going to follow this.
Well, my recommendation is follow it, because it's good for you. These are all machine learned models that tell you exactly what you should be eating. So I think you should be strict with your diet and you should follow exactly the Viome recommendations. And when it says minimize, then it's okay to eat that item maybe, once every few days. If it says avoid, just do your best to avoid it. And those foods that are enjoy and indulge, just eat them every day. It's not that hard.
The gut test is really, really good. What other things, exciting things you're working on now that people can look forward to?
Yeah, so this is our first clinically validated test. We have just clinically validated another test, which is the food sensitivity test, which is going to become available soon to everyone. Another test that I'm really particularly excited is we're actually moving into the human side. So we have developed a finger prick blood-based test that measures the physiological activity of thousands of genes in every single person just with a simple blood test.
So this is like a 23andMe type of thing, does a DNA, but that is just a SNPs, right? So what's the difference between 23andMe, what you're talking about?
Yeah, so remember, 23andMe analyzes DNA. So if they analyze your DNA today, when you have no chronic disease, or a month from now when you have some major IBD flare-up, the DNA is going to be identical. It doesn't change. However, we measure gene expression. So we quantify the expression of thousands of human genes in that blood sample. And so we can actually monitor over time and across populations how gene expression is correlated with health and disease, and how our gene expression is actually regulated by those gut microbes and by our foods, so that we can fine-tune human physiology using these environmental factors that are currently magic.
Momo, I'm just really fascinated here. So everyone always say that genes are your destiny. And what you're really telling me, my genes never change. It is my gene expression is my destiny. And there's no one out there really measuring the gene expression. So this is going to be completely unbelievable test where we will know what our human genes are doing in addition to what our microbes are doing in our gut.
That's right. So to truly understand the human body, we have to look at it as an ecosystem. We have 20,000 of our own genes, and we have at least that many microbial genes. And with the two Viome tests, one on the gut microbiome side and one on the blood side, we'll be able to understand both the human side and the microbiome side and the interplay between the two, so that we can fine-tune human physiology from that.
And will you be recommending the diet that we just fine-tuned?
Absolutely. So remember, our diet regulates not just the gut microbiome activities, but it also regulates our own gene expression. And then the gut microbiome activities regulate our gene expression. And so it's this interplay between those three systems that really determines our physiology. And that's what, for example, causes all of these flares in all of these chronic diseases that we currently don't understand. But with the molecular data that Viome will be obtaining, we will be able to understand the interplay of these and make personalized diet recommendations that prevent chronic diseases. And we will demonstrate that in clinical trials.
This is unbelievable, Momo. This has been fun, educational session.